Today I want to talk to you about enzymes. So you're going to come across enzymes in your chapter 4 reading, and they seem to fit a little bit better with proteins because they actually are proteins, and you covered proteins in your macromolecules. So I wanted to go ahead and introduce them here, so when you do get to your chapter 4, it won't be a surprise as to where they fit inside of the whole grand picture. So, enzymes. So, what are enzymes? Um, Enzymes are basically proteins. And remember, when we're talking about proteins or we're talking about macromolecules, that it's all about structure, it's all about the folding, it's all about conformation. And so when we have proteins, we're talking about first, the conformation. We have sheets and we have tertiary and quaternary structures, and that's just, based on how complex the folding is. So enzymes are proteins which are tertiary and quaternary structures. And enzymes act as catalysts. They accelerate a reaction. However, they are not changed in the process of accelerating that reaction. That's really important to remember. So they're very specific. Enzymes are kind of like a lock and a key. They're very specific for what they will catalyze. So one specific enzyme won't catalyze a product or a substrate that it's not very specific for. Just like a lock and key, you can have a lock, but only a certain key fits that lock. So it's the same thing with an enzyme and its substrate. Enzymes are reusable. And also, to try to identify an enzyme based on terminology, there is some terminology that goes into enzymes, and basically they end in ASE. So sucrase, it's an enzyme; lactase, an enzyme; maltase, an enzyme, signified by that ending of ASE. So let's look deeper at how enzymes function. So enzymes basically, they are lowering the energy of activation that's needed for a reaction to occur. So here, when you look at this graph, what it's showing here is a look at free energy, and you have your reactants, and your reactants basically go to products, right? And without an enzyme, you have this this free energy activation curve. Think of it as a mountain, a hill that has to be climbed in order for the reaction to proceed to a product. And so, as you can see, without an enzyme, that energy of activation is quite high. But with an enzyme, we can see, if you follow my cursor, it's much lower. And so the process can proceed faster. So I want to show you a quick video that explains this a little bit better.
Remember that in a chemical reaction, the reactants interact to form a product. In this graph, we are examining the relationship between the amount of energy in a chemical reaction and the time it takes the reaction to form a product. As you are probably aware, chemical reactions are rarely spontaneous. This is because of an energy speed bump that occurs during the chemical reaction. This speed bump is called the energy of activation, and it represents an energy threshold that must be overcome before the product may be formed. There is a relationship between the energy of activation and the rate of the reaction. The greater the amount of activation energy, the slower the speed of the reaction. However, cellular processes often require that chemical reactions occur at an accelerated pace. To increase the speed of a chemical reaction, cells use catalysts called enzymes. Enzymes function by lowering the amount of activation energy that is needed in the reaction, thus reducing the level of the energy speed bump in the reaction. Since less energy is required, the reaction may occur at a faster rate. Notice that the enzyme does not influence the energy level of the reactants or the products, but only the amount of energy that is required during the process of the chemical reaction. Without the use of enzymes, many of our body's processes, such as digestion and the processing of nerve impulses, would simply occur too slowly. In other words, enzymes make life possible.
Okay, great. So you can see enzymes are really important because without them, the reactions that our bodies need to actually perform, it would take too long. And so enzymes help to speed up this process by lowering the activation energy. All right, so let's look at what an enzyme actually looks like. So there's some terminology that you have to know or understand. So the substance that an enzyme acts on is called the substrate. So this is the enzyme's substrate. And here we can see that this enzyme in the illustration has a certain shape. And that shape fits the substrate just like the lock and key that we talked about before. And so we call this region where the substrate fits in a restricted region. So it's the restricted region of an enzyme. And this is what binds to the substrate. And exactly where it binds is called the active site. So what you want to remember is that enzymes have an active site. And that active site is conformationally specific to that substrate. More terminology. So you get this change in shape of an enzyme's active site when the substrate actually binds. And so I want to show you a movie that illustrates that a little bit better.
Sucrase is an example of how enzymes function in the body. Sucrase resides on the surface of the microvilli of the villi in the mucous membrane surface of the small intestine. This animation illustrates the way sucrase catalyzes the hydrolysis of sucrose, commonly known as table sugar, into its components, monosaccharides glucose and fructose. Hydrolysis happens when the sucrose molecule binds to the active site of the enzyme. The enzyme's configuration changes so that the oxygen bridge between the two monosaccharides is exposed to water molecules. The water molecule can then access the bond between the glucose and fructose sugars. The bond is broken by the insertion of the water molecule, and the disaccharide, sucrose, is split into two separate sugars. The enzyme configuration changes shape, and the two sugars are released. The site becomes available for another sucrose molecule to bind, change the enzyme's configuration, and be hydrolyzed. This process can be repeated many times during the lifetime of the enzyme.
So the enzyme is not used up in that process. It can be used over and over to repeat the same reaction multiple times. But what you want to notice is you saw that change in the shape of the enzyme when the substrate actually fit into the active site. The enzyme basically clamped onto it, and then the reaction occurred, and then it opened up and released it. So let's look at some factors that actually affect enzyme activity, and this is really important. So environmental conditions can affect enzyme activity. Things like extremes of temperature, pH, we'll look at that a little bit closer. Cofactors and coenzymes can affect enzyme activity, and enzyme inhibitors can affect enzyme activity. So three factors can affect how an enzyme actually works or whether it'll even work at all. Environmental conditions, cofactors, coenzymes, and enzyme inhibitors. Let's look a little bit closer at those things. So environmental conditions, this you need to know. So extreme temperatures are actually the most dangerous for an enzyme, and we use this knowledge because every time we learn something in science, we use it for the betterment, hopefully, of mankind, right? And womankind, humankind, to help us all. So you actually use this knowledge every day without even thinking about it. So extreme temperatures, high temperatures can denature or unfold the enzyme, completely unravel it, just as any other protein. If I don't want an enzyme to work, basically I can use a very high temperature. We use this in cooking, right? We don't want the enzymes in different bacteria to work, right? So we basically cook the food and it completely denatures, unfolds their enzymes, unfolds their proteins, because remember, enzymes are special proteins. So these factors also are the same that would affect proteins. So if I want to denature a protein, high temperature, right? Extremes of pH, number two, would also affect or denature a protein. When I talk about extremes of pH, I mean that we're looking at extremely acidic, pH of 2, pH of 1, of course pH of 0. Also on the other end, extremely alkaline pHs, so pH of 8, 9, 10, 14, will also denature proteins, right? And lastly, ionic concentrations such as salt ions can denature proteins. So I want you to remember when you're doing your studying for not your exam that you're having on Wednesday, but for your exam 2, enzymes will be on, and denaturing proteins will be on that exam. So make sure that you understand if I want to denature a protein, I can use extremes of temperature, so very high temperatures. If I use very low temperatures, let's think about that a little bit, right? Because we have a freezer. When you think about that, it will slow down the enzymes, the enzymatic action. However, it won't completely denature it. It has to be extremely cold, temperatures that are much colder than you have in the typical freezer in a home, right? And we use that to make our foods last longer so the enzymes don't break them down. But as you know, when you take them out and you let them thaw out on the counter, they will start to break down, which means those enzymes weren't completely denatured because they start to activate again, right? So extremes of temperature, especially high temperature, and extremes of pH, either extremely acidic or extremely alkaline, will denature proteins and denature enzymes. Let's look at cofactors and coenzymes. So inorganic substances such as zinc and iron, and also vitamins, are sometimes needed for proper enzymatic activity. So they're called cofactors, right? And this is one of the reasons why we need vitamins, because some of our enzymes actually need to have these vitamin cofactors in order to work properly. And so let's take a look at inhibiting enzymes so that they don't work. Not denaturing them, but inhibiting them. What does that mean? So there are two examples of enzyme inhibitors. One is you can have what's called competitive inhibitors. And so these are chemicals that resemble the enzyme's normal substrate, and they compete with that substrate for the active site. So they, as you can see here in the image in the cartoon, here's your substrate that should fit in this enzyme, and here I have a competitive inhibitor that has the exact same shape, and it's going to fit into that active site, and now the substrate can't bind, and so it has inhibited that enzymatic action. So that's a competitive inhibitor. But you also can have a non-competitive inhibitor, and these are inhibitors that don't enter the active site, but they bind to another part of the enzyme, causing the enzyme to change its shape, which in turn will alter the active site. So here, for example, when we look at this illustration, we have a non-competitive inhibitor that is bound to the opposite side of where the active site is. And we can see the active site here. And because that non-competitive inhibitor bound, it actually changed the shape of the enzyme, which changed the shape of the active site. And now the substrate cannot bind. So that's a non-competitive inhibitor. But let's look at why that's important. So the knowledge of these inhibitors we actually use, as we always do in science, to help the betterment of humankind. And let's look at some drugs, some medications that are actually enzyme inhibitors. So we have ketoconazole, it's an antifungal. So there are sometimes conditions where people will get, say, just really bad fungal conditions of the nails of the feet or the hands. Sometimes they're put on ketoconazole to try to stop that fungal condition. HIV treatments, some of them, such as this one called Kaletra, is actually an enzyme inhibitor. And one that you guys will probably all know because you've heard of it, Viagra, is actually an enzyme inhibitor. But we also take that knowledge of inhibitors and again, we try to use it and we make medications and medicines from it. So there are some plant toxins that are actually inhibitors, very powerful inhibitors. One is from the plant that you see here called Atropa belladonna, commonly known as belladonna. Also, deadly nightshade is another name for the plant. And we get the medication, the drug atropine, from this plant, and it's actually a toxin that the plant uses. And we use that, it's an inhibitor, and we use that all the time in emergency medicine. What it does is it speeds up the heart rate. So if you have a patient whose heart is just slowing down and threatening to stop, before it stops, you can actually give an injection of atropine and speed up the heart rate and save the patient. So it's always in an emergency crash cart in the emergency room. Also, atropine is used as a treatment for some of the nerve toxins that are out there that have been used. For example, there was recently on the political scene, there was someone who was running against the current leader in Russia, and he was poisoned, almost died. Turns out it was a neurotoxin that was used, and atropine is the antidote, and so he was given atropine. So a lifesaver, just the knowledge of the inhibitors and how they work has made such an incredible drug. In addition, chemotherapeutics, some plant toxins, are inhibitors. For example, there is one that is from the Pacific yew, or also called the Western yew, Taxus brevifolia, and a chemotherapeutic called Taxol. Paclitaxel is the full name for it. It's called Taxol as well. It's made from the Pacific yew, and that's used in breast cancer, lung cancer treatment, and also in ovarian cancer treatment. So this has been a huge lifesaver for many individuals. So knowledge of enzymes and how they work and how their inhibitors work is really important. And so now you know more about enzymes.